How about in the soils of our agroecosystems? Because we know we've destroyed the biology there with all the management protocols we've had, from the plowing to the biocides. Can we do this just by bringing the biology back into the system? First of all, we need to understand how nature is set up. There's a ladder of succession from a bare soil parent material up to an old-growth forest. And in that ladder of succession, we see increase in productivity. There's about 90 grams of dry biomass per square meter produced in a desert, compared to an old-growth forest at about 2,200 grams of dry biomass per square meter per year. As you see these changes, you also notice that we go from bacterial dominant systems to fungal dominant systems. This is very critical that we start to bring this back in our agricultural system, because where we are right now in agriculture, we're in a very bacterial dominant system. This is where we need to be, up here at about 1-to-1 fungal to bacterial ratio or higher. What we see as you increase fungal biomass, you see standing crop biomass increase in these systems. The bacteria, if you're bacterial dominant, it's negative correlation. So the more bacteria you have, the lower the productivity in these systems. We saw one of the biggest problems was the salinity on the dairy manure, and we moved the salinity down from 44 millisiemens down to less than 2 to 3. And it's also a vermicomposting process. The worms are part of this. But most importantly, it's a fungal dominant product that you end up with, and very biologically diverse.
Where we are in the Soundless Valley, we knew that a couple of the dominant features of this landscape are that it's arid, incredibly arid, and you saw that coming through. Those of you who live here know what that's like, and intensely cold in the wintertime. So we get down to negative 25, negative 40 at night, commonly for multiple nights in a row in the middle of winter here. So when I met David and Wei Chen and started chatting with them in 2015 about the static pile process that they had developed, and trying to understand what was it that they were seeing when using their compost in fields and in pot trials, it sounded intriguing, but there were a lot of hurdles for us to figure out how do we do this in our own environment, because it's not Las Cruces, it's not where they live, it's somewhere else, and the amount of sweat and effort and labor and toil that looked like it took to make one bioreactor did not look enticing for making lots and lots of them. So we were trying to figure out, is there some other way that we could try?
This reactor was designed and built by David and Wei Chen Sue Johnson in a project with USDA to develop a method that could easily and effectively compost dairy manure. This type of composting is a static composting process that allows the pile to be undisturbed for the complete duration of the composting process. This lack of disturbance allows the fungal communities to flourish and provides a more optimal process to break down beginning biomass substrates and produce a better quality compost. Besides for producing a more fungally dominant compost, the reactor process had also demonstrated a reduction in product salinity relative to the salinity of the beginning substrate.
The figuring out what the starting point would be involved John Kretzinger here and myself putting together a small batch using a 1-ton tote sack that was ventilated in John's hoop house over the span of a winter in 2017. And when we got results from that looked really promising for fungal to bacterial ratios, this was a very small batch, just one tote, like a half tote bag effectively. I guess it was 2 half tote bags. We sent it off for fungal to bacterial analysis. It looked promising. Gave it to one farm to try it out. They put it out on a field and put it on cover crops. And there was a distinct green strip that stood up and in the middle of that cover crop mix that really stood out as being very productive and drought tolerant compared to everything that was planted around it. So we just had a small batch, had one green strip that was through the middle of the field. So that was an early warning sign that, hey, there might be something to this. We should probably look at how to get this into a bigger project. Everything came together in fall of 2018. So that's what you're looking at now. Is it was put into this facility in fall 2018. The mix that we used, because it happened to be the stuff that we had access to, was cattails that were baled up after the season got dry, some cereal straw, we had some sheep manure accessible, and we had wood chips. So those were all mixed together. We used a mixture that we were shooting for 70% moisture at the time we were loading it in, and we anticipated that if we didn't get it right for moisture the first time, we were never going to get it right. So Dave and his team spent a lot of time making sure that the stuff that was coming in was the right moisture for what we were going to be attempting, because you'll see it in a moment, the surface is fully perforated, and there was a watering system over the top of it prior to this. When we turned that on, the water would all flow down those chimney tubes and run out the bottom, even without being able to infiltrate very far across. So if we hadn't have gotten it wet and right the first time, it would have been a nightmare to try to get it wet anytime after that. So that first step of getting it wet and then maintaining moisture was what we were trying for. The tubes that we used as forms, you'll see them in the background. They were all lined up over vent holes, which we're standing on top of, over a palleting system. And those tubes were lined in here at 1 foot spacings. Every foot, any which way, there was an air hole up and down to allow this pile to be actively breathing. And those are the things that we were thinking about early on in this process. The mix that we used initially, we intentionally pushed towards a relatively low carbon to nitrogen ratio, because we wanted to make sure it would go through the heating cycle early on, that it wouldn't there wouldn't be any question as to whether we zap the potential human pathogens in the starting mix. And so when it came in here, it came in hot and stayed hot for many days into the early process. And then eventually, within the span of 3 or 4 weeks, was cooled down to modest temperatures of they're about 80 or 85 degrees Fahrenheit. And it was then that we introduced a slug of red wiggler composting worms around the outside edge to make it into a vermicompost, or a modified vermicompost. And then from then on, it was just adding water. That was one of the things that David had clued us in on early, was if you're going to make a pile and if it's a stinking mess, day 1, day 2, day 3, it's probably not right. And if it's attracting flies, it's probably not right. At the end of the process, even though by a year and a half in, which is the point we're at now, there was enough aeration happening that it hadn't turned anaerobic even at the base. Our temperatures were 140, 150 or thereabouts in the early few days, through the pile top to bottom. And then it began to cool gradually. In a static pile, by the National Organic Program and the Food Safety Modernization Act, both, the requirement is that you can sustain critical temperatures between 135 and 170 degrees Fahrenheit for 3 days. So that allowed us to get through the critical phase of zapping any potential human pathogens. If you were in an actively aerated windrow type compost, it's a different rule, because you're reintroducing all those surfaces and mixing them all around. You need 15 days sustained killing temperatures, and so that's what most commercial compost will have to endure. Lots of turning, lots of labor and machinery hours, but also lots of opportunity to make a very bacterial-driven system, because there is so much turning.
So, who did your testing?
So we used 2 commercial labs. We took samples at multiple different time points through this process. Ward Labs did phospholipid fatty acid analysis on this material, and Earthfort Labs did a light microscopy analysis on this material. Analysis showed generally a trend of more and more fungi, more and more total fungal biomass was accumulating, and that seems to be because we could tell by just smelling the material. It smelled much more earthy or closer to mushrooms than anything else.
Yeah, those tests are about $800
Right.
a sample.
Okay.
So that's what we'll be looking at. We'll be comparing this also to multiple compost all over the U.S. that are being made this way. So we can see how predictable is this process in its end product. And of course, then we'll see on the other end, its application, how predictable is its influence on plant growth and starting to regenerate the life back into these soils.
Do you know the type of fungi that you're looking for in soil, or search
That's
is to decide which type of fungi are the most effective?
that's part of the search, to try to understand how this system works.
Good.
You know, it's not that nature's going to choose the same ones every time, because there's redundancy in these microbes. They have several talents. Nature doesn't always use the same one in the same place, at least what we've seen so far.
Fascinating.
But having that, as I say, a roster to choose from, the talent to choose from, seems to make this system function much better, more profitable for farmers, more productive as well.
The deliveries now will go out in spring. So we had an additional 5 months of resting time here.
We started out at 400 pounds per acre application. We were been successful in Wilcox, Arizona on a corn crop at 2 pounds per acre. 2 pounds per acre. We replaced 140 pounds of nitrogen with 2 pounds
Wow.
of microbes.
Now, did you dilute that with water to
Yeah,
apply it?
well, you have to make an extract out of this in order to apply it to there. And we injected it in the furrow with the corn that we planted.
So as you were
So when
planting,
you make your extract concentrate, how much extract does it take to make enough for 20 gallons to the acre with my corn planter when I plant my corn? Is it, do you add another 10 gallons of water to 1 gallon?
What we'll do is the ratio would end up to be 2 pounds of compost per 20 gallons of water. You'll be injecting 20 gallons per acre. Okay, that gives you your 2 pounds per acre rate. And this was with Howard Buffett,
Right,
and he was wanting to know what this would do for his corn. And we did that comparison. And with no fertilizer application, we only suffered a 6.6% crop loss. So he was profitable. What we're trying to relay is the soils that we have right now are broken. We know that. And we know what we've lost initially is the fungal community. I put it this morning, if you wanted to start a war on somebody, what's the first two things you take out? Logistics and communication. That's what the fungi do in the soil. So at that 2 pounds per acre rate, we're seeing for every square foot about 2 million bacteria and probably about 5,000 to 15,000 fungal spores that we're putting in with that seed. Replacing that biology is key to us to regenerate our agricultural systems.
Aerobic, it is 100%. It is 100% aerobic. So that means that the air infiltration is very important. So you don't want to have any time that having those air shafts collapse prematurely and then creating anaerobic condition. And so then also 70% moisture content is very, very important, because the worms and the microbes, especially fungi, they love that 70% moisture content. But you also don't want to oversaturate, because think about it, if it's oversaturate, you're impeding the airflow and they will drown, right? So these are living organisms. So you got to think about the concept of they need air to breathe, appropriate amount of air to breathe. They need an appropriate amount of moisture, but you also don't want them to dry it out. Whatever you do, do not let it dry out or freeze. Yes. Put in the red wigglers after thermophilic phase. And once the temperature comes down about 80 degrees Fahrenheit, then it's safe to put the worms in there, so you won't be cooking the worms.
This can replace about 170 pounds of nitrogen per acre. That was first year.
How did you apply it?
We made an extract out of it. I have an extractor online that I made. Poor boy, it's just a 55-gallon drum, a 5-gallon pail, and an agricultural pump. We just recirculated it and rinsed off this material through a screen. Then we took that and we injected it in as we planted the corn. If you have a fertilizer injection, liquid fertilizer injection system, you can use that and just inject in with the corn seed or whatever you're going to plant. Actually, 2 pounds of the compost mixed in 20 gallons of water, because most people can do 20 gallons per acre injection. Right. That works with your equipment, but 2 pounds of this per 20 gallons. It may take a little more of this because there's a lot of wood chips in here. It may take from 4 to 6 pounds per acre of this. But in systems where you don't have the wood chips, where it's all just this paste, that's all it takes.
We put on 400 pounds of this broadcast. What can we expect off of that?
Patrick,
It all depends on the number of organisms that you have.
yeah, we've
there.
got to analyze this first
Yeah.
to see what the microbial community, what the count is. So now we're trying to get about 2 million bacteria per square foot in our application. That's where we were successful in Wilcox, but we have to see what this has. We'll be looking at this in about 10 days.
If what's in this liquid extract is bacteria and fungi, if you put it on a field, do you need to feed it at all?
Well, okay, I'm glad you asked that.
Yeah.
She was asking if you need to feed these microbes. Yes. This is 2% of the solution. The other 98% is having a plant growing all the time to feed these microbes. It's the energy that keeps this system going. Just like us, without energy, we're third world. It's the same in the soil.
So living roots.
Yeah, living roots all the
is the way
time.
you should feed it.
If you have no living roots when you put it in, like you're putting it on a completely fallow field and you're seeding, say you're seeding on a fallow field, you're gonna hope that root is going to
If you put this in
do
with that
its
seed,
thing quickly enough to feed the microbiology
Yeah.
of the soils.
yeah, most of what I'm seeing here, the fungi are sporulated. So they're very stable. They'll take heat, cold, dryness, but it's when the conditions get right, that's when they start. And if there's a seed there to start working with that biology and pumping that carbon to feed those microbes, this system will start to regenerate. Like if you have a child, you have to feed that child.
Yeah.
And that's what we need to do. This is the child.
So could you put humate in with
This is packed with humates.
it? So it's gonna be packed with humate. Okay.
If you grab a sample and smear it with your fingers,
That's what I'm
and that dark,
that's
it doesn't rinse off, that's your humates.
It stains your hand.
Yeah, like that.
It's hard to wash it off.
So yeah, they're producing humic.
okay.
So is there any economic advantage to get the soils regenerated faster by doing multiple years of application of your
Yeah.
bean?
Oh, on this, if you're doing 2 pounds per acre,
Yeah.
it needs to be done every time you plant.
Every
Every
planting.
time you plant?
Either that, or if you irrigate, you can put it in an irrigation system.
And if you are doing a dry land farming in a more brittle environment, yes, most certainly you need to reapply every planting.
Every time you plant
Yeah.
Yes.
in the arid west.
Yeah.
Yeah.
How about where we broadcast the compost? Do we have to redo that every year too?
If you put it at 400 pounds, that's what I started with, was 400 pounds. But from that point on, look, I applied once, and I just had crop after crop after crop growing. I've never had to replace it.
No.
When you bring this biology back, you increase your photosynthetic capacity of that soil. You increase the carbon use efficiency in that soil as well. All this plays together. They just start stacking on each other. What I did in the experiment in the greenhouse in order to rule out any extraneous things is I used the same different ratios of sand and compost to achieve that bacterial dominant to fungal dominant. So I'm using the same communities all the way through, just increasing amounts.
Right.
And that's where I saw this increase in productivity, that 5 times increase in net primary productivity. That's possible as you move these soils from where we are right now, which is bacterial dominant and low carbon, to a soil that's high carbon and fungal dominant. And that showed me that 5 times increase. And now I've done it 3 times in the field with different crops. and seen the same thing, a 5 times increase in that productivity. So we're pretty confident that you can increase a poor soil from what it could produce. So that will start helping open up those soils. If you get this biology back in there, allow you to get air back in there, and that water will start to flow through your system instead of being ponded.
This pond is terrible.
See, it opens that system up. We're just trying to mimic what the cattle did on the Great Plains. If not everybody wants a cow around, and cows always get where you don't want them. So, but this kind of replaces that.
Yeah, so regarding food safety, the GAP qualifications, this qualifies as a compost. And so same idea. If you were making a compost tea, if you were making a compost extract, actually compost extract, because there's no intentional booming of microbes in a tank, it fits better in our potato production system than a compost tea, because compost tea you're required to send off samples periodically and have records of samples of the compost tea product. Compost extract, you've got to show that the compost itself went through the critical temperatures, and then you're good to go.
What if you don't use manures?
Still, yeah, the GAP certification requirements are going to have you providing temperature logs, or at least having a paper copy of that. Same as when you're using compost on the soil.
What test is going to give us the kind of biggest bang for our buck?
You did ask that one.
I know, I'm sorry. Have to ask it.
That's the toughest one. We're still.
Yeah.
Okay, I'm going to start out by saying this. Any one of us are not going to be able to tell the state of your health by going to a doctor with one test.
Right.
So how do you assess your soil health with just one test?
So
And the
over time-
answer is no.
Yeah, but over time, but if you're doing PLFA, which we've talked about somewhat maybe inconclusive.
Well, PLFA.
And yes, go on.
PLFA is not good for a fungal to bacterial ratio. The designers of the test even say that.
No.
What they say it's good at is change over time.
Right.
Now, determining or defining what that change needs to be is still an unknown in science, because this is so dynamic. Just a regular soil test with nutrient availability is not going to tell you, because as I showed in the presentation, what's available.
It's not about the nutrients that was
No.
really powerful in terms of plant growth.
It was about the biology.
Yeah.
That's,
What about CN ratio?
uh,
Would that be more helpful?
I can't
Change
tell
over
you.
time, maybe?
I haven't done that one. So I've been concentrating in the metagenomics and metatranscriptomics to see, can we find a marker microbe or a set of marker microbes that lets us know, okay, our soil is healthy because it has these microbes in this ratio. That's what we're really looking
So,
for.
so it sounds like the science is still really emerging in
Oh.
terms of
We're at the very
DNA
beginning.
and RNA analysis.
This is at the very
Yeah.
beginning. And if it wasn't for the Human Genome Project, we wouldn't be where we are on this.
Right.
It was their understanding and studies of the human microbiome that has led to this.
So what we were talking with a microbiologist from CSU, was that yesterday? Yesterday. Yeah, and we had talked about, doing a really good baseline and taking some of that soil and freeze drying it.
It's best, if you're going to think about metagenome and metatranscriptome, you need to have your sample put into what we call RNA later. It's a stabilizer. That can be frozen at minus 70 or minus 80 degrees, and it's good for decades. And that allows you to come back at any time. And it's also good for PLFA. If you're going to do a PLFA, because PLFA really needs to be frozen as quickly, or cooled and chilled and gotten to the lab the next day. If you can't do that, RNA later will allow you to do that. But there's so much we don't know about this.
Right.
And we have some great tools now. I can tell you about 2,700 species in there. I don't know what they do. I know what some of them do. But it was the quorum sensing part that really popped up. As you improve your soils, you see those microbes work together as a community and express genes that they couldn't as a single individual, but as a community they can. And that's where we saw the growth, the plant growth.
But as far as right now for the soil test, we feel the metagenomic and metatranscriptomic test is still the most reliable to kind of seeing what microbial communities are there, and then referring back to your net primary production. But that is more expensive because it takes probably about $1,000 for analysis per test sample. And also you need to have somebody with expertise like David in able to understand what you're reading.
Anybody can look through a microscope. They can figure out how to use it, or
But it's
go
understanding
take a class.
what you were looking at. And then the experience really come into play.
You start to build up your reserves in your bank. And that's when this system starts working for you. Build up enough eventually that you'll have money for venture capital, money for these organisms to go out and do something else.
Turns
And-
into interest.
Yes.
Yeah.
So the crop growth is an interest accruance. So you put the money into the soil as savings, and then that builds up as equity, and then what you get back is interest in what you sell from your crop.
And this is all about energy. It's that energy capture and how it's utilized, how efficiently it's utilized. You're the kind we learn from.
Yes.
You know, you're willing to do something different. And observation is key in all this. It's never going to be the same year by year. It's not an easy button. It's not a formula-driven way to farm or ranch.
It's very much adaptive.
It's
And
you're
without
observing.
observation, you cannot adapt.
I got here late. Another question I have, how did you increase the fungal activity in this? I mean,
What we're seeing is, since it's a static process, it's not like a windrow compost,
Compost.
where you're turning it. That destroys that whole community, that fungal community. Where this, it's undisturbed, so it allows that fungal community to grow. It's 70% moisture content, so that's the ideal
for fungal.
fungal. And it's also aerobic. Since fungi are eukaryotes, they utilize oxygen in their
If
respiration.
it goes anaerobic and you disturb it, you're not going to increase the fungal community.